a todos y a todas. Les damos la bienvenida a este simposio número 10 que tendremos en el día de hoy. Es un simposio de ALACF en el que vamos a hablar sobre obesidad, una enfermedad sistémica. Tenemos 4 muy importantes presentadores que nos van a estar compartiendo sus charlas en el día de hoy. Queremos agradecer, por supuesto, a la organización de tanto de SAFIS como de ALACF por la invitación a participar de esta mesa. Y bueno, vamos a dar comienzo, así tenemos el tiempo para poder escucharlos. Mi nombre es Carolina Canifi, voy a estar coordinando esta mesa. El coordinador es el doctor Pedro Leme. Él también va a ser quien presente la primera charla del día de hoy. Él es Associate Professor de la Federal University of Rio de Janeiro, Brasil, y él nos va a hablar de deterioro de la función pulmonar durante la obesidad. Así que, Pedro, va a ser un gusto, te escuchamos.
Okay. Thank you very much, Carolina, for the introduction. I will share my screen full. Okay, so thank you very much for the ALACF to accept the suggestion of this symposium. So my talk is about the lung function impairment. So I prepare agenda talking about the epidemiology, the respiratory system and immune system, more immune system related to the respiratory system associated with obesity. And then at the final section, I'll talk about the diaphragm and the alterations due to obesity. So we know that obesity is considered a pandemic since 1975 to 2014, according to this Bluher published in Nature Review of 2019, the obesity is increased around the world. There are a lot of factors, social, cultural factors, local environment, even body size preferences. But the main, I would say, and not good factor is the susceptibility to food marketing. The prevalence of obesity in Brazil and also in Argentina, I looked at yesterday. In Brazil, the prevalence is 22.3%, and in Argentina is a little bit higher, is 27-28%, according to the last global database from World Health Organization. So there are a lot of causes of obesity. It's very difficult to put in a single formula, but usually they share the same criteria. You have a huge food intake for several factors, and there are a lack of physical activity due to muscle pain, joint pain, low fitness level, and emotional barriers. A lot of factors contribute to the lack of physical activity. When we associate the respiratory system with obesity, we can see a decrease in lung volumes. One very famous lung volume is actually a capacity, it's called the functional residual capacity. So we can compare the open symbols that are patients without obesity and the full losango symbols that are patients with obesity. There is a decrease the functional residual capacity according to the BMI. And the reasons are related to the huge extrathoracic fat volume and even the interthoracic fat volume due to enlargement of big vessels, and the lungs pay all this kind of bill, decreasing the volume inside. So there are a lot of consequences about this reduction in lung volume. The most, I would say, complication is the distal airway closure that may predispose these patients to airway hyperresponsiveness. And we associate the adipose tissue with immune system cells more related to the pulmonary environment. With a lean subjects, we have a production, a constant production of adiponectin, and adiponectin can promote the differentiation of M1 phenotype macrophage to a M2 phenotype macrophage. A macrophage more related to the resolutive phase of some infection or something that may damage the lung. In lean subjects, you expect a low level of leptin production. But when you have an obese patient usually the levels of adiponectin is decreased and the levels of leptin is increased. So leptin can promote the oxidative burst of neutrophils, for example, can improve their survival. They can act longer at the membrane, alveolar capillary membrane. Leptin can decrease the V-cadherin. V-cadherin is a protein connecting endothelial cells, for example. They are very important to not allow the plasma starvation from the vascular side to the epithelial side. And even leptin can increase the adhesion molecules that may interact with leukocytes and promote lung injury. But one question remains very, paradoxically in the literature. During critical illness, what happened? So the clinical literature about obesity and ICU admission, for example, due to respiratory causes, they are very paradoxically. For example, in this study of Lorenzo Bal published in 2017, there is a reduction in mortality according to the BMI. Usually, you would expect an increase in observed mortality according to the BMI, but several studies show that this is not observed. And even Zhao published in PLOS ONE with some similar studies and adding new ones, they also show that obesity has a kind of protection due to respiratory infection, respiratory impairment. This kind of obesity paradox was triggered before pandemic, but during pandemic, there is this publication of Elizabeth Williamson from Great Britain. They show in 23 million patients, and then the analytic population was 17 million patients. And she showed to us that the class 1 obesity is not a factor of mortality according to the COVID-19. Only started a good correlation with BMI around 35 to 40 kilograms per meter square. So this means that the class 2 and class 3, they are associated with mortality, but not class 1. This is difficult to explain. During critical illness, one additional author, Richard Dana, published that in a range of BMI between 30 and 40, there is a decrease in mortality. You see, the BMI in 25 to 30 is around 22% of mortality. And then in a BMI of 30 to 40, there is a decrease to 13% of mortality. And then then BMI higher than 40, yes, there is a huge increase in mortality. So what are the reasons for this paradoxical result? So the authors, they started to rank some factors. Some of them were discussed even before the pandemic. But here we can see that the obese patient, they have a metabolic reserve during a catabolic state that usually is the ICU admission and ICU enrollment during increased activity of renin-angiotensin system, which may paradoxically protect against hemodynamic instability, and preconditional chronic pro-inflammatory states, which may train immune cells to this kind of infection or inflammation in the lungs. This publication, recent publication, showed to us in a BMI of 30 that neutrophils from COVID patients, they are activated by the expression of CD64, for example, but when they analyzed different cluster of differentiation like CD11B, CD65, and CD162 that are related to the neutrophils migration, they are not increased in COVID-19, severe COVID-19 patient with a BMI of 30. So this may explain a little bit this paradoxical result that the neutrophils are activated, but they do not migrate very well. So this may somehow protect the respiratory impairment in obese patients. So our group was very interested to associate obesity with anesthetics, because usually the amount of surgery is increasing, and there are a lot of possibilities to conduct your anesthesia plan. So there are inhalation anesthetics, there are intravenous anesthetics, and we were interested about the propofol, because there were some previous literature showing to us that propofol was there was a kind of protection of propofol in alveolar capillary membrane. So this was the master degree of Luciana. Luciana is an anesthetist. She performed experimental obesity by giving the experimental diet, which is similar in terms of calories with the conventional diet, but we changed the proportion of proteins and carbohydrates. She showed to us that obese lungs and obese adipose tissue, they express more IL-6. This was to corroborate the literature. But when we analyzed the interaction between propofol and obesity, we observed a lot of distal airways with this configuration. And then we measured the airway resistance, and it was really high after propofol infusion, but not with barbiturics and dexamethasone, which is a new option in the OR nowadays. So we were not satisfied with this result. We want to understand what's going on with the commercial propofol, because the commercial propofol that we buy and we promote the anesthesia of these patients is not only the active component. We have egg phospholipids, we have soybean oil, we have glycerol. So we try to dissociate the commercial propofol with the lipid excipient and only the active compound. So we primarily extract the macrophage and neutrophils from bronchoalveolar lavage, lung tissue, and adipose tissue. We expose these cells to saline, lipid excipient, active component, and the commercial formula, which is actually the sum of lipid excipient and active component. We expose these cells to 50 micromolar for 60 minutes. Usually a very simple surgery is around 60 to 80 minutes. And what we observe? We observe that neutrophils show the lower migration when exposed to the commercial propofol and also the lipid excipient, but not with the active component. So we may expect that neutrophils are impairment due to the commercial because of this lipid excipient. So this is actually, we have to thank the laboratory which gave to us, and we can promote a better lipid excipient to not cause this kind of alteration in neutrophils. We observe mainly in lung tissue this kind of alteration, not in adipose tissue, although we can see here a kind of similar behavior, but they did not achieve significant difference. So the last part, I would like to talk about the diaphragm. Why study the diaphragm? Diaphragm in rest condition is a very nice muscle, respiratory muscle. It has some kind of advantage because it consumes less than 30% of the global VO2. It requires less than 5% of cardiac output. So there is a good weather for the diaphragm because diaphragm can perform almost the totality of respiratory work. But in a condition of shock, distributive shock, cardiogenic shock, we can see that the diaphragm now show its real face. It consumes 50% of the global VO2. There is a high necessity of the cardiac output. So the weather is completely different in a condition of shock. That's why we need to sedate and paralyze the patient, because it will steal the oxygen from other organs, for example. So what the literature was telling us that about the peripheral skeletal muscles in obesity, previous papers showed to us that there are a presence of large lipid droplets in the muscle. there was a kind of shift from glycolytic to oxidative muscles fiber phenotype, a decreased expression of PGC1 alpha. PGC1 alpha is a transcript factor associated to the biogenesis of mitochondria, and increased generation of reactive oxygen species. And these muscle cells, they are more prone, predisposition to damage. So what about the diaphragm? Very few publications about diaphragm and obesity. So this was the master degree of Gisele Rodrigues. Gisele Rodrigues performed a different obesity model. It's not the diet model performed by Luciana. Here is the restriction model, in which we decrease the amount of male pups per den by 3 male pups per den in the 3rd day or after birth. And the control animals, they stay with 10 pups per den, usually is around, like physiologically is around this, 11 to 12. And there is after 21 days, there is this kind of a restriction about the pups, and then 150, you have the excrement. We observe the diaphragm, an increase in the exclusion of the diaphragm, an increase in the expiratory thickening, and an increase in the diaphragm thickening fraction. Here we can see that the exclusion in obesity is higher than the control, and the thickening, you can see here in the muscle, not a very good resolution, but we can see that we can observe this alteration at the diaphragm, at the ultrasound. We were interested about the mitochondria function during obesity. So we removed the diaphragm, we used some solution to promote the permeabilization of this fiber, and we did some kind of challenge in order to better understand what's going on with this fiber. So the first challenge was with pyruvate malate and succinate. We observed that by activating the Krebs cycle, we did not observe difference between obese and control animals. When we introduced ADP, in order to study the couple respiration coupled to protons, we observed that the obesity has a couple respiration higher than lean animals. And then when we introduce oligomycin, we can analyze the uncoupled respiration here. We did not observe any difference between control and between obesity and control animals. But when we introduce FCCP, we can measure the maximal respiration, even higher than the physiological range. It's a kind of a huge challenge to really see if this fiber can breathe at very strong condition. So FCCP, we observe by the very permeabilization to protons, we observe that obesity is higher than lean animals. And here we can see after KCN introduction that we can measure the residual respiration. There were no differences between obesity and control animals. Okay. So look at the fibers. We observe a shift between from glycolytic. We can see here the myosin heavy chain 2B, a decrease in obese animals, and an increase of myosin heavy chain 2A in obese. So there is a kind of interconversion, mainly here in the intermediate fibers. And we also analyze the fatty synthase, PGC1 alpha, and SOD1, SOD2. We observe an increase in fatty synthase. We can promote, which can increase the substrate introduction in this Krebs cycle. PGC1 alpha was lower at obese patient, which we can, which suggests that the biogenesis of this fiber is reduced. We did not observe difference SOD1 and a decrease in SOD2. SOD1 is superoxide dismutase, more related to the cytoplasm, but SOD2 is more specific to the mitochondria functional. So we can observe that obese, there is a reduction in SOD2, which we can observe that if there is a decrease in SOD2, superoxide may promote more deleterious effect in this fiber. So the suggested mechanism is that during obesity, there is a kind of overload. The diaphragm can become thick over time. There is a reduction in glycolytic fiber and an increase in oxidative fiber. Due to reduction in SOD2 and PGC1 alpha, maybe the superoxide, they can increase their activity. We did not, unfortunately, measure the superoxide in this fiber, but we observed the consequence of all these suggested mechanisms. That was the senescence of phenotype of mitochondria. So we look at the ultrastructure and observe very different mitochondria in obese compared to control. So the final message is that obesity is considered a pandemic disease. We are living a pandemic, not now, but obesity is here for a long time ago. It seems that obesity paradox is not broken during COVID-19. This is very difficult to explain. There are some authors that do not agree with this term, paradox of obesity. The use of anesthetic agents in obese population may have an impact in immune cells. Now, we observe that propofol do not show a very good conversation with obesity. Obese diaphragm may have changes in muscles fiber phenotype, followed by increased mitochondrial respiratory capability. And if this kind of condition may predispose to oxidative injury and early senescence of these fibers. So I would like to thank you all the students from our lab and the professors that I work with here at the Institute of Biophysics Carlos Chagas Filho at the Federal University of Rio. Their financial support, and thank you very much. This is our group in Rio de Janeiro. Thank you.
Thank you, Pedro. It was a very interesting presentation and obviously on a very trendy topic. So thank you very much. I have a question. I don't know, maybe another question could be on chat, but I have a question because I think that we might expect that obese people to have less skeletal muscle mass. And how can this fact and maybe also the release of myokines from muscle, how can this fact affect respiratory function?
A decrease in the peripheral muscle, right?
Yes.
When we talk about the respiratory muscles, they had a kind of increase in the oxidative function, but over time there is a kind of a lower, for example, expression of some lactate receptors. So they are more prone to fatigue and respiratory failure in a short term compared to the lean subject. So if there is a kind of infection in a patient without obesity and with obesity, so the obesity are more prone to go to the ICU than a lean subject. They do not have some kind of because of this early senescence, the respiratory muscles is different and cannot support a huge demand of oxygen and CO2 elimination. So usually they are more prone to go to the ICU. What is interesting is this obesity paradox, because in the ICU, they have a good, how can I say, clinical outcome compared with patients without obesity. This is something that we need to better understand, because as I said, that there are some epidemiological authors that do not agree with this kind of paradox obesity. They tell us that this is a kind of error that we are propagating with this kind of obesity paradox.
Okay, thank you, Pedro. Bueno, no hay preguntas por el momento en el chat. Vamos a continuar entonces con la siguiente presentación. Pedro, te dejo para que la puedas introducir. Si llega a aparecer alguna pregunta, después podemos realizarla.
Okay, thank you very much, Carolina. So I introduce the next speaker, is Professor Ana Paula Couto Dável. She's an associate professor at the Universidade Estadual de Campinas. So it's our great pleasure to have you here, Ana Paula. Thank you very much.
Thank you, Pedro, and thank you, Carolina, for this introduction. Can you see my screen?
Yes.
Okay. Just I close something here. So thank you very much. It's a pleasure to be here. I would like to thank the Sociedade Argentina of Fisiologia, a Sociedade Brasileira de Fisiologia, e ALACF, for this opportunity. And today I'm gonna talk about the modulation of vascular function by perivascular adipose tissue. I'm Ana Paula Dável, as Pedro said. I'm associate professor at the University of Campinas, and if you want to know more about our work, here our site and social media. So it's well known that the perivascular adipose tissue, also called PVAT, is a fat surrounding most of our arteries. And this fat is composed by mainly adipocytes, but also by other cells like endothelial cells and immune cells. And PVAT has gained attention after the discovery of its physiological anticontractile function. So, as you can see here in this study, the authors investigate the vasoconstrictor response in response to angiotensin II, serotonin, and phenylephrine. And you can see in black symbols the constrictor response without PVAT, and in open symbols the constriction in the presence of PVAT. So it's clear that in the presence of PVAT, we have a reduction in the vasoconstrictor response, suggesting that this fat can limit the vasoconstriction. So, and this function of PVAT seems to be dependent of the release of factors from PVAT. As you can see here, when we transfer bath solution from a aorta with fat to a vessel without fat, we can still see a vasodilator response, as you can see here in this record. In aorta without fat, when we add the solution from fat from another vessel, we can see a reduction in the constriction. So nowadays, we know that there are many factors derived from PVAT that has been identified as adipokine and nitric oxide, and these factors are released by the PVAT, and these factors can act directly to the smooth muscle cells, inducing vasodilation, but also can act through the endothelium, so can induce endothelium-dependent effects. And these effects are mainly seen in healthy arteries, as mean in physiological situations. So does the obesity impact PVAT-derived vasodilator factors and the PVAT anticontractile function? So this question was addressed several years ago by a group using human very obese individuals with severe obesity. So they isolate small arteries, resistance arteries from these individuals, and these arteries were studied without PVAT here in blue and with PVAT here in red. So when they look at the vasoconstrictor response to noradrenaline, they observe that in these very obese individuals, there are no anticontractile effects of PVAT, suggesting for the first time that human obesity can be associated with PVAT dysfunction. So next, these authors investigate the same protocol, now investigate these individuals 6 months after bariatric surgery, as mean after losing weight. And in this situation, the anticontractile function of PVAT was restored and comparable to health PVAT, as mean comparable to lean individuals. So severe obesity is associated with PVAT dysfunction. And as you know that severe obesity is a cardiovascular risk factor, pivot dysfunction can be a pathological mechanism linking severe obesity and cardiovascular disease. So far, I have shown you that pivot has an anticontractive function in healthy arteries and that severe obesity can impair anticontractive function of pivot. So we raised the question that, is there a sexual dimorphism in pivot function in both health and in the setting of obesity? So, first of all, does sex difference matter for obesity? If you take a look in the numbers around the world, so Pedro just showed the pandemic of obesity, but if you take a look by sex, we will see that we have more women around the world overweighted and obese. And if you take a look here in Latin America, you see that sex difference is really important. So we have here data from 80s in continued life, and the numbers of men and women obese, but look at the numbers in the prevalence in women. We have almost 40% of the population obese. So we need to take a look in this. So a postdoc from our lab, Jamara Victorio, has looked at the literature to try to find studies showing pivot-derived factors in males and females. And what she found, that was not surprising, that most studies were conducted in males, and most of factors that we know so far were identified in pivot from males. But we are started to study this pivot in females, and we have an idea that this pivot can have also an anticontractive function and secrete some factors. However, sex difference in pivot secretory profile is poorly understood. So in her postdoc, Jamara aimed to evaluate the anticontractive pivot function in resistance arteries of male and female mice that fed a controlled diet or a high-fat diet as a model of obesity. So Jamara took male and females that were fed 3 months with both control and high-fat diet, and in the end, she evaluated some parameters, and both sex presented higher body weight, increased perigonadal fat, hyperglycemia, increased plasma insulin without change in blood pressure. So we modeled obesity, similar obesity model in both sexes. Next, she tried to understand the vessel function. So she isolated mesenteric vascular bed, as you can see here. And from this bed, she isolated resistance arteries. And these resistance arteries were placed in our organ bath without adjacent pivot, as you can see here. And also some arteries were kept with the adjacent fat. So in these two situations, we evaluated the contractile response to the alpha-adrenergic agonist phenylephrine. So as first results, we observed the data in males. So as you can see here, the contractile response to phenylephrine, and as expected in control mice, the presence of perivascular adipose tissue reduced this contraction in blue symbols. However, when we evaluate semi-parameter in obese male mice, the pivot anticontractile function was not altered. As you can see here, the potency to phenylephrine was reduced by pivot in both groups. So in male mice, with this diet, the pivot function was not altered. However, when we look at females first, in control situation, the effect of pivot was higher, was bigger than in control mice. As you can see here, the anticontractile effect was pronounced. However, in obese female mice, this anticontractile function was lost. So we have a really impairment of the pivot effect, as you can see here, the reduction in the potential to phenylephrine in control animals, and no significant reduction in this potency in obese female mice. So this data suggests for us, for the first time, that female sex may have a susceptibility to pivot-mediated vascular complications in the setting of obesity. So our next question was, what are the mechanisms involved in this sex difference observed by for pivot dysfunction in obesity? So we take a look in the emerging role of aldosterone and mineralocorticoid receptor activation in the cardiovascular damage in female obesity. And why aldosterone and mineralocorticoid receptor? So going back to 90s, we have this a work, these studies in obese individuals, and they take a look in the circulating hormones in women and men obese. And they observed that both sexes have hyperinsulinemia, increased activity of renin, but so interestingly, they observed that only obese women has an increased circulation levels of aldosterone, what was not observed in men. And also, they observed that this higher circulating level of aldosterone positively correlated with increased visceral adipose tissue area, showing for the first time that maybe aldosterone plays a role in sex difference in obesity. So we know that aldosterone can act through the activation of mineralocorticoid receptors, DMR. We know that in addition to be expressed in tubular cells in kidney, MR is also expressed in vascular cells and adipocytes. And we have a bunch of studies that described that in obesity, we have that the hyperlipidemia in males can act through inducing sympathetic activity that mediates hypertension and vascular dysfunction, but in females, looks like this hyperlipidemia has another mechanism through the activation of aldosterone synthase in adrenal glands, and this induces increased circulating level of aldosterone that mediates cardiovascular damage. So we asked the question, it's possible that this pathway, this increased aldosterone signaling can be involved in the pivot dysfunction in female obesity. We took a look first in the endothelial cells, and in summary, we and other groups have shown that when we have an overactivation of MR in endothelial cells, we can have oxidative stress by an increased production of ROS, a reduction in endothelial nitric oxide synthase activity expression, which leads to decrease in NO bioavailability and production. And we also have an pro-inflammatory signaling by induction of adhesion molecules in these endothelial cells. Interesting, we and others have shown that these effects of overexpression of MR are more pronounced in female sex in response to risk factors, including obesity and high diabetes. And our group also has previously shown that pivot can express aldosterone synthase and MR and can produce significant amount of aldosterone. But so far, we don't know this signaling in obesity in pivot. So our second question in this study from Jamaria was, could aldosterone and MR signaling be involved in the sex difference of pivot dysfunction in obesity? So for this, she took male and females that were fed with controlled high-fat diet, but in the last month of feeding, they were treated with spironolactone, that is an MR antagonist. And in the end, we have the 6 groups: control obese and obese plus spironolactone, males and females. And then she studied the anticontractile effect of pivettes in mesenteric arteries. So as a result of this part, as expected, we observed an anticontractile function of pivettes in males here in blue and in females here in red. This effect was also seen in male obese mice. However, as I said before, this pivette function was impaired in female obesity. And when the animals were treated with spironolactone, the similar response was observed in male. However, in females, we can see a restore of the pivette anticontractile function, suggesting for the first time that maybe MR signaling is involved in the pivette dysfunction in female obesity. To take a look if the MR signaling is activating the pivettes, we isolated the pivette from these vessels, and as you can see here, we observed that in the setting of obesity in female mice, we have an increased expression of aldosterone synthase, an increased expression of this kinase, ISGK1, that's activated by this pathway, and also an increased expression of the beta subunit of the sodium channel in neck. And spironolactone in females was able to prevent these changes. No changes were observed in this signaling in obese males. So finally, as we know that pivette inflammation is a mechanism of endothelial dysfunction, when we have immune cells infiltration, increased release of pro-inflammatory cytokines, we have a reduction in NO bioavailability. We evaluate immune cells profiles by flow cytometry in the pivettes from male and female mice with obesity. And here we have the results in females. In females, we have an increased number of macrophages and M1 macrophages, inflammatory macrophages in pivettes of female obese, and also increased number of dendritic cells in these tissues. And as you can see, spironolactone can reverse this infiltration. And finally, we measure nitric oxide levels in this perivascular adipose tissue. And as you can see here in female obese mice, we have a reduction in the fluorescence to this NO probe. And here we can see this quantification, a reduction in NO levels that is restored by spironolactone. So as conclusion, I would like to say to you that our results suggest that in female obesity, pivette has a upregulated MR signaling that leads to a reduction in nitric oxide levels and also a reduction in anticontractile function of pivettes associated with an immune cell infiltration. And these effects were prevented by MR antagonist spironolactone. So MR signaling seems to contribute to pivette dysfunction and immune cell infiltration in obese females, but not in males. So this study reinforced the importance of investigating sex-specific treatments and pharmacological targets for obesity-induced vascular complications. So I'd like to thank our laboratory team, especially Jamaria Victorio, that made the most experiments that I show you today, and also our collaborators and you for your attention. Thank you very much. Gracias.
Thank you very much, Ana Paula, for your great talk. This is very important issue to study the sex-specific difference in treatment and the pathophysiology. This is a very good, up-to-date issue that we should pursue. Yeah? I have a very short question, Ana Paula. Did you check the hemodynamic of these animals? They have the same like amount of sodium, and because this is a chronic model for obesity, right? And a very short one. Thank you very much.
Okay, thank you, Pedro. We have the plasma to measure sodium and potassium, but we still don't have. We need to measure, especially because spiral can change this electrolysis, but so far we don't have.
Yes. Okay, thank you. Carolina?
Yes, we have a question from Paola, Paola Casanello, so I want to read the question. The question is, you show sex differences in contractile response to phenylephrine. Is the vasodilator response in these vessels also regulated by the PVAT? Is the question.
Yes, Paola, thank you. Very important question. Yes, I didn't have time to show, but the PVAT can also modulate this vasodilatory function. Yes, it's true.
Well, I think that we can continue with the next presentation, Pedro, if you are okay.
Yes.
Well, thank you, Ana Paula, for your great presentation. Bueno, ahora vamos a escuchar a la doctora Paola Casanello, que es profesora asociada de la Facultad de Medicina de la Pontificia Universidad Católica de Chile, de Santiago de Chile, y el tema de su charla es el origen de la obesidad. Así que, Paola, con gusto te escuchamos ahora.
Microphone, okay. Gracias, Carolina. Thank you very much, Carolina and Pedro, for the invitation to present in this symposium. I'm very happy to share with you. My talk is oriented to the beginning of life, so the early origins of obesity in women with obesity and opportunity for intervention. So, as has been already shown by the other speakers in this afternoon, there is a high prevalence of obesity worldwide, and this is a great concern, especially in our country. Pedro showed before Brazil and Argentina, but Chile has over 30% of the population with obesity in adults. And we also have a lot of obesity in children, we're the highest in the region, really. In the OECD countries, we are the highest country with overweight and obesity, reaching around 74% of our population. So we have national health surveys performed every 3 or 4 years. And the last one we had showed that we have 38% of our population, women, 38% of women in our country over 15 years have obesity. So, and the levels of morbid obesity have been increasing in the last years as well. So this means that 1 in every 3 women begins their pregnancy with obesity. She has her first pregnancy prenatal control, and she is already obese. So what is the problem with maternal obesity? It has a lot of health problems for the women during pregnancy. These women gain more weight than what we would expect. They have a higher rate, very much higher rate of gestational diabetes, and also preeclampsia, and a lot of other gestational complications, preterm delivery, and breech problems in the delivery, C-sections, etc. They also have the fetuses of the moms with obesity also have a lot of different kinds of malformations, fetal death, excess fetal growth. So we have macrosomia in the fetuses, they grow bigger. And also during the neonatal period, we have a lot of intensive care unit need in these children, cerebral palsy, and all kinds of neonatal complications, which haven't been very much described in this population. And then we have a lot of metabolic syndrome and child obesity as well. So in our group, we have been interested for some time already to look at what's happening so early in pregnancy that the women, the children of these women have the higher risks of being obese themselves when they are children. And there's a lot of people trying to look how early is this obesity stated, and they have been looking, is it the maternal obesity, or maybe it's the maternal weight gain during pregnancy, gestational weight gain. So in this very nice paper published in PLOS Medicine, where is a meta-analysis of individuals, more than 60,000 individuals of different studies, and they put them together, and they recognize that here's the maternal body mass index, and you can see here the higher the maternal body mass index at the beginning of pregnancy is, the higher risk, this is the relative risk, or the odds ratio chart, sorry, for the child overweight and obesity in early childhood, mid-childhood, or late childhood. And you can see over BMI of 30, this risk is severely or significantly increased in these children from early, but it still perpetuates in the adolescence. So in this paper, they describe that maybe 20 to 40% of the childhood overweight and obesity prevalence could be attributed to maternal overweight and obesity together at the beginning of pregnancy, whereas 10 to maybe 20% could be attributed to excessive gestational weight gain. So during the weight the mother gains during pregnancy. In this meta-analysis done a few years ago in different animal models of obesity, it is very clear that there's a lot of long-term effects in the offspring of women with obesity. And in this paper, particularly, they show, for example, what has to do with body composition. So body weight, body fat, and fat mass in the offspring of the mothers with obesity is significantly increased. So there's a higher risk for this, is very significant. Also systolic blood pressure in these animals, and also a lot of lipid profile alterations like triglycerides, cholesterol, and LDLs, and clearly insulin resistance and the risk of high glucose, high insulin, and high insulin resistant markers in the offspring after birth and in the first times of development, so childhood equivalent to humans. How does maternal obesity confer risk of chronic disease to their offspring? Well, we have to think maternal obesity is, in strictly speaking, an adult with obesity. And as all adults with obesity, there's a chronic low-grade inflammation. We've talked a lot about the adipose tissue in the vascular tissue and in the affecting respiratory function, and also a lot of metabolic alterations due to this low-grade inflammation. Pregnancy itself so overimposes a lot of adaptations, cardiovascular and metabolic adaptations, leading to a maternal inflammatory, systemic inflammatory condition during pregnancy, which is related to some of the adverse effects I just showed you before. And this leads to placental inflammation. We cannot forget that the placentas, they are mediating the interaction between the fetus and the mother. So there's a lot of evidence showing that placenta itself is producing a lot of pro-inflammatory molecules, as well as leptin, for example, regulating oxygen transport and increasing oxidative stress in these children, impairing several placental functions, including nutrient transport to the fetus, which is very much increased due to these inflammatory conditions, which leads also to very early markers of inflammation and increased adipose tissue and programming hyperphagia in the children, appetite dysregulation, and all kinds of inflammatory conditions in the fetus in utero life. This is one of the first papers more than 15 years ago, showing how maternal pre-gravid BMI, for example, here they used overweight and obesity, so BMI over 25 versus women with BMI under 25, and they showed that the offspring of these women had altered a lean body mass, so there's no difference in the lean body mass, but there's a increased fat mass and body fat percentage in the offspring of these women, although there were not differences in the birth weight. And they also, when the child is born, almost all the adipose tissue is under the skin, so it can be quantified by skin folds. It is very used, and a lot of skin folds determinations have been included into the formulas to estimate body fat, and they are very reliable. And they studied different skin folds all over the body of the fetus, and they could see that most of them were significantly increased in the offspring of women with overweight and obesity. Very recently in this paper, they showed that this fat mass is very directly related to maternal BMI. So higher maternal BMI, the higher fat mass in the offspring. But this is only true if the mom is obese. When the moms have normal weight, this correlation is not present. What are the proposed mechanisms for this increased fetal adiposity in this woman? Well, what's happening is the placenta has a whole lot of transporters regulating nutrients. One of them is the GLUT1, which is the most abundant exose transporter in the placenta, in the maternal and the fetal side, which is pushing all the glucose from the mother to cross to the fetus. We know that women with obesity have higher glucose levels independent of diabetes. They're not over the norm, but they're slightly higher. I'm going to show you later our data that show this. And this glucose is going through the placenta freely because these transporters are not regulated. The glucose goes freely to the fetus. This increased levels of glucose in the fetus induce the liberation of insulin in the pancreas, and also they are regulating adipogenesis in the liver. There's also an increased lipogenesis and liberation of triacylglycerides, which are going to the adipose tissue, activating LPL in the membrane of the adipose cells at the adipocytes, and generating an accumulation of adipose lipids in the cells. So this will be mainly the main mechanisms regulating the accumulation of fat in the adipose cells, but also fatty acids can cross in a regulated form and also freely the placenta, and these non-esterified fatty acids are also contributing to this increased accumulation of lipids in the adipocytes. We published earlier this year when a cohort of children born from women normal weight and obese, that the whole group, when you put them all together, most of them, the children with obesity, of moms with obesity, had higher levels of adiponectin. And these higher levels of adiponectin, we didn't know because when in adults, adiponectin, in obese adults, adiponectin is negatively regulated. So obese people have lower levels of adiponectin, which is a very important insulin sensitizer and regulator of the metabolism. So we didn't know what happened early. If these children accumulate a lot of fat, would this adiponectin be also downregulated? But we found there was a controversy in the literature, and we find that this adiponectin is increased and that the males have more adiponectin than the females. So, and what is very clear from the children of women with obesity, that the male offspring are bigger. So they have a higher growth in utero compared to females. But when you go to look at other markers like leptin, there's an increased levels of leptin in the umbilical cord of the children born to women with obesity. But this increase is mainly conditioned by the female offspring. So the female offspring are regulating more the metabolic part, and the male offspring are more regulating the anthropometric growth in utero. We also looked at the insulin, and we didn't see differences between the groups. It has been also very clearly shown how the offspring of women with obesity have altered immune cells and immune function. For example, in this study, they measured the levels of interleukin-6 in the offspring of women with normal weight, overweight and obese, and they found in the umbilical cord high levels of interleukin-6. They also stimulated these cells with LPS to see the TNF and IL-6 secretion. And what they found is that the offspring of women with obesity, so the white bars, they have lower capacity to secrete IL-6 and also TNF-alpha in response to LPS. So although they are more pro-inflammatory in the basal, when you stimulate them, they are less, they secrete less cytokines in response to LPS stimulation. And finally, in a later paper of the same group, they showed that not only these monocytes have lower capacity to secrete cytokines, but they also have a less capacity. The offspring of women with obesity, the monocytes in the umbilical cord at birth, have a lower capacity to migrate in response to a stimuli compared to the lean, the offspring of lean moms. Here's the adult monocytes as a control. So many things have been thought, what kind of interventions can be done during pregnancy to avoid all these adaptations. And one opportunity is inducing physical activity. And lots of people have tried doing interventions with physical activity in women during pregnancy when they have obesity, and they have failed. The mothers do not adhere to physical activity, and it has very little effects no effect on birth size of the fetus, and no effect on the long term either. So some dietary interventions have been studied. And one of the opportunities we had was to collaborate with a group of epidemiologists in the University of Chile, which were studying the effects of supplementation with docosahexaenoic acid, DHA, which is mainly present in fish and algae. Some seeds as well have it, and being supplemented during pregnancy. So instead of having to elongate and desaturate the chains of the fatty acids, and during the processing of the metabolism of the precursors, we are giving the pregnant women the DHA, the polyunsaturated fatty acids, directly on capsules during the day. The last meta-analysis that has been done regarding DHA supplementation during pregnancy have shown very clearly that there's a reduction of preterm labor and very preterm birth, less than 34 weeks during pregnancy. It also decreases the incidence of low birth weight and the requirement of neonatal intensive care unit, or the dying or being very sick of the neonates. It also increases pregnancy length in a few days, and also these children grow more healthy and they develop more healthy. So in this, what will happen when the mother is taking the DHA during pregnancy? Well, this is the schematics of the placenta. Here we have the syncytial trophoblast, which is the cell that is regulating the transport of nutrients between the mother and the fetus. So the polyunsaturated fatty acids, or fatty acids in general on the maternal side, are metabolized, and also they are transported through different transport proteins to the fetal side of the placenta. The placenta itself also has a lot of consumption of fatty acids. It stores them, they activate signaling pathways, and they generate mediators as eicosanoids. These fatty acids will cross to the fetal side, and how much do they cross? It was studied by a group some years ago, where they studied saturated, monounsaturated, and polyunsaturated fatty acids in the maternal plasma and in the cord plasma. And they showed very clearly that there was no difference in the saturated fatty acids or the monounsaturated, but the polyunsaturated fatty acids were very clearly accumulated in the fetal side of the placenta. So the mother is mainly taking the fatty acids from her diet, or in this case from the supplementation, and passing them to the fetus in a preferential manner. In our group, we have been, due to this, to the interest in the nutrition during pregnancy and the PUFA supplementation, we have been studying, I'm only going to show you part of the data. We've been studying growth and adiposity in the infants. We're following these kids up. They're now around 3 years old. We've also studied a lot of markers in the placenta, some B12, folate metabolism, inflammatory markers, fatty acid transport and metabolism. We've been looking at immune cells in the umbilical cord, and also some progenitor cells, and we've been interested in vascular function in the arteries. I'm going to show you today some data of the EpiFat study that we set up with the children of these mothers that were supplemented with DHA during pregnancy in a randomized controlled trial, where some of them received 200 milligrams of DHA, which every woman should receive. It's like eating 2 portions of fish per week, or 800 milligrams of DHA per day. And we also had a normal weight control group for the basic science mainly, and for the reference values. We evaluated the moms during gestation, anthropometry, and some morbid data. We have samples of these ladies biobanked, and we also did some food frequency questionnaires. At the delivery, when the neonates were born, we measured the kids, we did adiposity calculations, we also took blood from the cord, isolated immune cells and placental tissue. And at 4 months, the children came with their parents to study and to be evaluated anthropometrically, and also to study adipokines and some blood analysis. What I can show you here, for example, the BMI, we have 3 groups then. The group with obesity that received 200 milligrams, so the normative amount of DHA, the basal amount that every pregnant woman should take. Women with obesity, so BMI over 30 at the beginning of pregnancy, took 800 milligrams, and the normal weight group. And we can see here very clearly that we had a little difference in the age of the patients between groups. This is part of the randomization, but between the groups is very interesting. You can see the BMI, of course, is the inclusion criteria. 34 is the mean BMI, but there was a lot of patients with over 40 of BMI. We have the normal weight group. Interesting to note between the groups is the fasting glycemia. As I showed you before, these women did not do gestational diabetes. We left them out of the study. The gestational, in this EpiFat follow-up study, not in the women's study, but in the children's study. And you can see here the glucose, the fasting glucose is around 82, 84, and the normal weight women have 78. So there's a significant differences in the basal glycemia. This is before starting the intervention. There was no difference in insulinoma. The insulin resistance was around 55% in the groups. The women did not know they were insulin resistant. And in the glycemia profile in the second trimester, you can see still there's a very important difference between the glucose levels in fasting and also post the post charge of 75 milligrams of glucose. In the children, when they were born, we can see some differences in the length. The children of the normal weight women were a little bit longer, and also the weight divided by height ratio, which some studies have shown is a good indicator of body composition. You can see is a little bit lower in the group of the normal weight and higher in the group of the obese women. Interesting, the mothers taking both of them, of the groups that took the PUFAs, the supplementation with the PUFAs, have lower triglycerides compared to the normal weight that did not take the PUFAs. The LDLs are higher compared to the control group, and the insulin in the cord was higher, significantly higher in both groups compared to the control as well. When we measured the body composition of these children at birth, when you look at the skin folds and you calculate the fat, the body fat, there was no difference between the 2 groups that were intervened, that received the intervention. And of course, they were all different. All the skin folds and the body fat were different in the control group compared to the both children of women with obesity, independent of the dose of the DHA. There was no difference between groups. So when you estimate in the regression analysis, you have 2 ways to look at this data. One is the effectivity. So if you compare all the children of the women that received 200 compared to 800, and you just study the differences independent of the number of capsules they took, if they adhered or not to the treatment, you just take them together. And when we look at them as effectivity, just comparing 200 versus 800, we do not see differences in the fat, the body fat. But when you look at the efficacy, let's say one of the outcomes was the DHA, and we measured the DHA in the umbilical cord. When you how you control by the DHA levels in the umbilical cord, it is very clear that the body fat and the fat mass are significantly decreased in the children of the women with the higher levels of DHA. So the efficacy is positively regulated by DHA, maternal DHA supplementation. We also looked at two markers, adipose in the kids, and also adiponectin, which is this very important insulin sensitizer. And you can see there's a lower ratio of subscapular by triceps skin fold. This is one of the ratios that is very well characterized in the children as an early marker of adiposity at 4 months, and also changes in trunk versus total skin folds is decreased in the group that received, whose mothers received 800 milligrams per day. And adiponectin is increased in this group, so they have more adiponectin, which is something we believe is something positive, so more insulin sensitizing hormones, and also the change between adiponectin at birth and at 4 months is also increased in the group that received the higher dose of the DHA. We later looked at some of the immune markers, and we looked at the monocytes in the umbilical cord from the 3 groups, the groups that received the low dose, so the normative amount compared to 800, and you can see IL-6, the messenger levels in the monocytes, together with MCP and TNF-alpha, all pro-inflammatory markers are increased in the adipose, the women with the offspring of women with obesity with the normative amount. And if the mother has a higher dose of DHA, these levels, these pro-inflammatory markers are clearly regulated for IL-6, MCP, and TNF-alpha. I'm not showing you all the data. We have some more genes. We looked at some immune regulators as PCG1-alpha, etc. Macarena Lepes, which did this part of the study, she wanted to know, independent of maternal supplementation, is DHA regulating the expression of these pro-inflammatory markers in monocytes? So she took a group of a pilot study of normal weight women and women with obesity that had not been supplemented with DHA. She took the monocytes in the umbilical cords and took them. This is the control, the LPS stimulation the last 4 hours, and she pre-incubated with DHA or an antagonist of the receptor of DHA, GPR 140, and she later stimulated with LPS. And what she found is that the LPS stimulation is, of course, increases the messenger of these pro-inflammatory cytokines, IL-6, MCP1, IL-10, which she did others, but I'm just showing you these ones. Although the induction was a lot lower in the monocytes of the women with obesity. This is very interesting. And in those that were pre-incubated with DHA, she did not see this increase in the expression of these messengers. So there's a very important immune modulation done by DHA on these fetal monocytes at birth. Parallel to this, we wanted to compare to see if these changes in the response and also in the basal inflammatory response, there was some epigenetic markers. So we took 2 cohorts, the cohorts of the MITE study, the one that I showed you just now, 200 and 800 milligrams of DHA in women with pregestational obesity. And we had done 1 year ago another study where we had studied normal weight and the offspring of women with normal weight and pregestational obesity without DHA supplementation. So we took these 4 groups. We had done epigenome, epic methylation array on the monocytes of the cord of these kids at birth. And then Cristina Silva did all the bioinformatic analysis of these children, and she compared a whole lot of groups, but now I'm going to show you just the comparison between the normal weight and the pregestational obesity group. And she can see there's a lot of hypermethylated probes, 21,000 probes are differentially methylated, hypomethylated, and a little less are hypermethylated. But as you can see, DHA, 200 milligrams and 800 milligrams are doing a huge change in the methylome in the monocytes. This has been described before in cord blood mononuclear cells as a whole, but this is the first time that we're showing this effect specifically on the monocytes, how maternal supplementation with DHA can change a huge amount of methylation sites in the mom. When we went to look where these differences were, we can see there's most of the changes when you compare obesity with normal weight mothers, the offspring, the monocytes of the offspring with obese and normal weight, they are mainly on the CPG islands. When we look at the differences,
Paola,
See?
We are without additional time. If you could,
Okay.
Yes, thank you.
I'm going to stop in the next 2 minutes. So there was changes mainly in the open C and the induced by DHA. Right here are some of the changes in the genes. We're interested in the PPRs, which we are now doing pyrosequencing to investigate. So adding all this together, we know that the offspring of women with pregestational obesity have higher risk of obesity. They have the DHA is efficient to decrease the total body fat and decrease subcutaneous fat. There's an increased expression of pro-inflammatory and immunomodulatory genes in the monocytes that were reverted by the supplementation with DHA. And we know that DHA supplementation in women with obesity regulate DNA methylation, especially in immunomodulatory genes and metabolic genes. I didn't show you that now. And these changes are mainly in CBG islands and open C. I didn't show you, but we've been looking at what could be the factor inducing this pro-inflammatory basal response. And we have been looking at maternal endotoxemia, which we have very recent data showing this could be present in these patients. This could be the trigger for the pro-inflammatory response in these progenitor cells. So we are believing that maternal supplementation with DHA, eventually previous, but also during early fetal development, could reduce the markers of the programming of these adipose and also the immune cells. We are looking at progenitor cells, and this is what I want to thank all the people in our group, the international collaborations, fundings, former students, and of course our collaborators, clinicals and basic science, and of course the team. So thank you very much for your attention, and I'm happy to have questions if there's any time.
Okay, well, thank you, Paola. It was a very interesting presentation too. I have a lot of questions, but we have no more time. I just want to prioritize questions from the audience. And we have a question from Paola Valero. She says, thank you, excellent presentation. These obese women who have a lower capacity to secrete cytokines in response to LPS, may they have a certain degree of immune suppression or a higher risk of acquiring infections? Is the question.
Yeah, I have to remind that this is happening in the fetuses. We haven't measured the cytokines on the moms. We have blood. We're looking forward to measure it, but in the fetuses, they have lower capacity to secrete cytokines in response to LPS. And yes, in fact, the offspring of women with obesity do more infections. And necrotizing enterocolitis and a lot of neonatal infections, yes.
Okay,
thank you. Well, Pedro, maybe
yes, let's go forward.
Okay.
Yes, I would like to introduce our last speaker, Professor Egberto Gaspar de Moura. Professor Egberto is a full professor of physiology and pathophysiology at the State University of Rio. It's my great pleasure to have you, Egberto. Thank you very much.
Ok, thanks Pedro, muchas gracias al Comité Científico de la Sociedad Argentina de Fisiología. As Pedro introduced me, I'm going to talk about understand the mechanism of obesity-related metabolic syndrome. The problem is that obesity is not a simple, complex one. And as you can see here, we have several thousands of factors that can affect our development and change our way to gain weight. And as already shown by Paola, Pedro, and Ana, there are several factors. I'm going to concentrate in this hormone here, the leptin, but also we are going to talk about all these things that can interact the intestinal GI, gastrointestinal tract, with what happens on our brain, and also the microbiome that is very important to regulate all these societies, and also the accumulation of body fat in our organism. As already shown, worldwide obesity has nearly tripled since 1975, as you can see here, and the growing in obesity is similar to women and men, boys and girls. And now obesity is considered a pandemic. In Brazil, this is very grave, very severe in the last 3 decades. We have an increase between 2006 and 2018, around 7% of the obesity index. We have around of 100 million Brazilians overweight. It's almost a double of all the Argentinian population. I see that the same occur in Argentina in terms of percentages, but in Brazil, the amount is very huge and has an impact in our public health system. And you can see here, around 57% of men in adult age and 54% of women are overweight. And there is no difference in genders, but more worry about this is that 50% of women at the reproductive age are overweight or obese. And the problem is this syndrome here, the metabolic syndrome, that with obesity, central or visceral obesity, you have a higher risk to develop diabetes mellitus, to develop cardiovascular disease, hypertension, hypertriglyceridemia, and also you have other things that not are defined as the metabolic syndrome, but are associated with metabolic syndrome as a high risk of Alzheimer's disease and Parkinson's disease, and also the high risk of cancer, breast colon, prostate, esophagus, and endometrium. So also we have a high risk of the new coronavirus infection, and also its complication as demonstrated by Pedro in the first presentation. And when we talk about obesity, we think about the main suspects: high dense calorie foods, genetics, sedentarism. And so there is one simple solution. Okay, they eat good food, nutrients as vegetables, and do exercise. But that's not so simple to achieve. And why that? Because there are more causes than that we see now, like the couch potato lifestyle, the ultra-processed foods. And we have to pay attention about the effect of endocrine disruptors has on these obesity pandemics, as well as the climate change and also the stress that we have living today, more than in the past. Around the 70s, the middle of the 70s, in the last century, we have a paper that changed all our understanding about the increasing of prevalence of obesity. And it was done by this couple, Mervyn Sousa and Zina Stein, that are from South Africa, but they are working at the Columbia University in New York. They have to fled from South Africa due to the apartheid, and they are anti-apartheid activists. And what they changed with Ravelli? They changed. They show that in a cohort of 300,000 19 years old recruits, which when they born, they were exposed in womb to the Dutch family in World War II. And it's interesting because those ones that have this malnutrition during the first half of pregnancy, they have a high prevalence of obesity. Later on, in a decade of 10 years ago, almost, there are several epidemiological studies conducted by the group of David Barker in England, and they show the same association of low birth weight and increased risk of obesity, insulin resistance, and dyslipidemia. And they propose the theory of the fetal origins of health and disease, or the thrift phenotype. And they have several books about that and publishes and continuing to publish several articles even after the death of David Barker. But there was a change of the concept by two important researchers in this field of the DOHD. That was Peter Gluckman and Mark Hanson. And they changed the name of fetal origins of adult disease for the name that we use today, developmental origins of health and disease. Mainly because all these factors did not act only during pregnancy, but through all other phases of development, such as the lactation, infants, and even adolescence. And they changed the names for fetal orange to DOHD, metabolic programming. And a metabolic programming is has a sense of determinism. That is not the case because it can change according our nutrition during life or exercise. And they change for developmental plasticity. And they propose another theory that in this book, that's the mismatch theory, that has as premise the following. Obesity is one of the most important challenges of public health, even more serious in developing countries, such as Brazil and Argentina. And it becomes worse as we become better in development. And that's why, because our adaptation mechanisms developed during the evolution of our species are no longer adaptive in this new environment which we live today, due to recent breadth and breast exchange. During evolution, we developed a system that allowed the mother to signal to the offspring how the environment that they are going to live is. And this signaling is through the placenta that Paola already showed to you, and also through lactation. Because in the milk, we have several micronutrients, but most important, several hormones and growth factors. And I want to call the attention for the leptin, this hormone that's produced in adipose tissue and goes through the milk. Microbial communities in the milk, and the microRNAs that has to be studied. So this predictive adaptation is already signaled by the mother, all the mammals, through the milk. So during evolution, we have our genetics that seems not so important to humans considering obesity, because we have only 200 cases in the world of monogenic disease associated with obesity. Not syndromic, of course. So we have changed our nutrition from the paleo diet, more health, to this not ultra-processed food diet that is unhealthy. More than this, we have different kinds of stress during our evolution, but we have one adaptation of the autonomic nervous system and also of the hypothalamic pituitary adrenal axis, like cortisol. And everything is adapted to allow that the mother had a good pregnancy. And she needs to accumulate fat. And that's why all the mechanism is doing to accumulate fat to provide a good nutrition for the fetus. So you have here the metabolic syndrome and the balance between the cortisol and the sympathetic nervous system, thyroid hormones, insulin, and leptin. And I'm going to show the importance of leptin because leptin that was almost 30 years ago was discovered, and it was a good hope for the obese persons because we think that you can inject leptin and then the obese person becomes thin. But that does not occur because after you administrate leptin, rapidly you have a leptin resistance. It only works if the person has a deficient in the leptin gene. So leptin is a hormone that has several actions. Pancreas, in our reproductive system, in the blood vessels, in our immune system, and also in the hypothalamus, decrease the food intake, but also controlling several hormones. Here you have the effect on the hypothalamus, but also you have that leptin acting on these neuropeptides that are anorexigenic or orexigenic. They increase the sympathetic activity and increase thermogenesis, but also the blood pressure. And when you inject leptin in excess, you have an increase of these signaling proteins, the SOX3, and this SOX3 inhibits the signaling pathway of leptin, but also inhibits the signaling pathway of insulin. That's why leptin resistance and insulin resistance comes always together. And also there is a problem because the resistance of leptin is tissue specific and temporally distinct. So first you have a resistance on the hypothalamus. So the person did not decrease the appetite when leptin is higher, but leptin continues to act on a sympathetic activation and causing hypertension. That's why the obese has a high risk to hypertension. So this programming effect that is related to the predictive adaptive response or development plasticity occurs during critical periods of life, as gestation or lactation, and is set by different imprint factors such as hormones, nutrition, environment, and through different epigenetic effect that was already talked by Ana Paula, like stone acetylation, DNA methylation, microRNA. And if the initial conditions are the same, you have a better adaptive response. But if you change drastically the initial conditions, and we changed that because we are living more, we are living 3 times more than our antecessors, you have a higher risk of disease. And which are these imprinting factors? Like nutrition, you have undernutrition or overnutrition, different nutrients that we use today, endocrine dysfunction like the gestational diabetes mellitus, hypothyroidism, the maternal obesity, as already was demonstrated by Paola, environmental factors, that's very important today with the plastics, herbicides, pesticides, infections that we are living now with the coronavirus, drugs as alcohol, cigarette smoking, cocaine, marijuana, and stress. Here I can show something that could be very similar to what we have now with the coronavirus, because during this period in Canada that have a great ice storm, there was a great period of isolation. And the children that had 6 years old when this occurs, when they were fetus, they have a higher risk to develop autism. So in our lab, we studied several of these imprint factors, but we start with the protein restriction, protein malnutrition. And we submit the mothers during lactation to protein restriction or energy restriction, and follow the offspring to adulthood. And you can see the energy restricted gain more weight and the protein restricted less weight. But interesting that during the weaning time, both groups, protein restrict or energy restrict, had hyperleptinemia. And the mothers also had higher hyperleptinemia. And probably the mother secrete more leptin through the milk, and is that leptin that passed to the pups. And when this offspring becomes adult, the offspring of control mothers, when we inject leptin to them, they respond well to leptin, decreasing food intake. But the offspring of the protein-restricted group or the energy-restricted group did not answer. So they have leptin resistance, and in this leptin resistance is due to a decrease of STAT3 on the hypothalamus.
Egberto?
No,
I'm sorry, but we are on time.
2 minutes.
Okay.
Okay, 2 minutes, please. We have other two models, the prolactin inhibition, and you can see here also in this model, you have an increase of leptin in the milk, and also increase of leptin in the serum of the weaning brat. And also with another model that we expose the mother to nicotine, you have the same. You have increase of leptin in the milk, and also an increase of leptin in the serum of the pups. So we decide to inject leptin in the pups, and we inject in the first 10 days of life. And these pups, when adults, develop higher food intake, higher body weight, hyperleptinemia. And look, this is interesting because 7 days after weaning, they have a peak of leptin, a peak of insulin, and also have hyperinsulinemia at adulthood, and hyperinsulinemia at adulthood, and hyperleptinemia. And also they develop leptin resistance. When we inject leptin, they didn't respond as well as the controls, and several other alterations of metabolic syndrome. So we have several studies, several models of different imprinting factors, and even with differences in a serum leptin concentration at lactation, all these models developing a hyperleptinemia at SEPT1, overfeeding, at adulthood with leptin resistance. And to finish, I'm showing here, one experiment, a recent experiment, studying the effect of the sympathetic nervous system on the brow adipose tissue. And as you can see, both male and females, they have a decrease of response when they have early weaning, or when they are exposed to cigarette smoke, the same, they have a decrease in the action of the sympathetic nervous system at the brown position. So if you want to see, we have 3 revisions about that, about the early weaning, about the tobacco smoking, and about the importance of breastfeeding that we talk about everything. So while acting insulin resistance is so evolutionary important to be naturally selected if it can cause a disease, is because it can provide during gestation glucose to the fetus. The main problem with programming studies that in nature, all these imprinting factors act together. So you have the endocrine disruptor, the ultra-processed food, the early weaning, the contamination of our environment, cigarette smoking, everything is acting together, but we isolate the factors to study in our experimental studies. So I want to thank the people on my lab, especially Patrícia Lisboa, Elane Oliveira, my postdocs, Luana, Rosiane, and Yala, and all the students that work with us. Thank you very much. Muchas gracias pela atenção.
Thank you very much, Professor Egberto. You know, we are run on time, but I would like to do a very short question, Egberto. What are your thoughts and what may be the next step if someone wants to study the brown adipose tissue? If you are looking at this very, I would say, very promised tissue or pathophysiology, what do you think about it?
Okay, we humans have a process of browning of our white adipose tissue. That's why it's so important to us, because several decades ago, the scientists think that brown adipose tissue has only in rodents and bears, but now we are very certain that in humans, white adipose tissue can be transformed to brown adipose tissue. And the contrary also can occur. So we have a small amount of brown adipose tissue that if we become obese, it transforms in white adipose tissue. So we have to develop new therapeutic strategies that can transform white adipose tissue in brown adipose tissue. That's
okay.
I think.
Very nice, Egberto. Thank you very much. So we are run on time. I would like to thank all the professors, Professor Ana Paula, Professor Paola, Professor Egberto, and also my co-chair, Carolina. Please,
thank you.
If you want to say some words, I would like to thank the ALACF for this nice symposium. Yes,
the same. I want to say just thank you to Safis and ALACF for this symposium.
Okay. Thank you very much. Bye-bye.